dear students, I hope all of you are very well and every day you are learning something new. My name is Abhijit Devnath. I am working as an assistant professor at NIT Pharmacy Institute, Greater Noida. Today, I am going to tell you about microbial biotransformations, how you can use microbes to transform something to a new product and how you can use good microbes for our good purposes to get our desired product. So, I am going to tell you about the introduction of microbial biotransformation, then I am going to tell you what are the types, advantages, disadvantages and few of the application. I am going to take another lecture with specifically highlighting the applications of microbial biotransformation. So, these words biodegradation or biotransformation or biocatalyst, these words are many of the time you might have heard, and these are the buzzwords which might be sometime created confusion in your mind, but you have to understand all the things are actually related with each other. So, if you have in case of new pathway or new enzyme, both the things can be happen, the biodegradation or biotransformation, okay, for to get improved biodegradability, reduction in waste product or process development. We can use some enzymes to degrade some product, so that we can get improve the biodegradability, or if you have some waste material, if you want to get some desired product from that waste material, in that case you can also use the microbes. Now, biocatalyst, what biocatalyst, how they can actually do. Now, what is biocatalyst? The modified substance or substrate range, reaction mechanism or mathematical or physical some description will be there with the biocatalyst, something enzyme. So, it will, if you have new target, it will transform, and also in alternatively process development of new enzyme is actually happening, and also in feasibility of desired reaction in case of our from catalyst to biodegradation or new reaction. So, this is how it is actually related with each other. Now, if I come to the definition, so biotransformation, it is a process by which organic compounds are transformed one form to another to reduce the persistence toxicity of the chemical compound. Right now, if you visit any of the waste recycle that is a activity or that is a organization, those are actually doing those are actually recycling or that is processing the waste material, you will be observing they are using lots of microbial enzymes or directly they are actually using some microbes, so that they can transform one form to another, so that toxicity can be reduced through of the chemical compound. Okay. If I tell more, this process is actually aided by some by major range of microorganism and their products. What are the microorganism? Bacteria, fungi and various kinds of enzymes were actually taking their help. Biotransformation can also be used to synthesize compounds of material if synthetic approach is actually challenging. Okay. If any synthetic approach is actually challenging, in that case we can also use biotransformation of very compounds. I hope you have listened the story about the penicillin. The great scientist called Alexander Fleming, he actually synthesized this thing called penicillin from Penicillium notatum, but it was actually not very easy to use orally on those days. After that, our scientists they have done lots of work, so that they can get some product, get some penicillin which can be taken orally with a very simple manner. They actually synthesized the new form of penicillin. Okay. But in every cases, whatever the good story was happened in penicillin, in every cases such good things is not going to happen. In such cases, what you have to do? In such cases, you have to take the help of the microbes, so that microbes can transform one substance to another if the synthesis is not very easy. Okay. Now, Natural transformation is actually very slow process and non-specific and less productive. But in case of our lab or our in microbial biotransformation, this microbial biotransformation or microbial biotechnology are gaining importance and extensively utilized to generate metabolites in bulk amount with more specificity. For example, alcohol, wine, how we are actually producing wine? Obviously, using the microorganism. Further, biotransformations are generally preferred to chemical reaction because of substrate specificity, stereospecificity and mixed reaction condition. Okay. For example, temperature, pH, pressure, etc. In such cases, we are actually preferring. If I tell you more, the environmental pollution due to biotransformation is almost insignificant or negligible. See, sometimes our environmentalists, they are very much concerned about the waste material or the byproduct or metabolites are developed by or are generated by various industries. But you have to understand, if any chemical actually get generated by some industry, and if we treat those chemicals with good microorganism and it may metabolize that thing, and this if the new metabolite is not harmful to our environment, then it will be a good thing. But if it is harmful to environment, then we can use another enzyme or another microorganism which will be metabolize that metabolite also. Okay. So, that is why environmental pollution is actually very that is low or negligible. In addition, it is very easy to apply recombinant DNA technology to make desired improvement in biotransformation. And another advantage of the biotransformation is that it is easy to scale up the process due to limited number of the reaction. Okay. This is the another advantage. Many times biotransformation is involved more than one type of reaction, and the conversion of time required for biotransformation is related to the type of reaction or the substrate concentration what are actually using. In general, oxidation, hydrolysis, dehydration reaction actually are completed within a few hours. These are the reaction which are actually happening or we are actually performing during biotransformation. Actually, we are actually using bacteria or fungi or other substances or sometimes enzymes, so that such reactions may happen and we may get our desired product. Now, sometimes actually get confused between the biotransformation or metabolism. I have already cleared at my first slide what are the difference between this thing called how you can use the biocatalyst or our bioremediation, how they are actually different. Here I am trying to tell you how biotransformation is slightly different than metabolism. In definition, if you just see the definition, the biochemical transformation taking place in a particular compound, drug or xenobiotics. In this case, total number of biochemical reaction taking place in living organism. Okay. A particular compound, only a particular compound will be get transformed, and here takes place a living organism, almost everything. Target only particular compound, and here whole organism actually involved, metabolism. This is how it is so much different. You should not be confused with the metabolism. Drug metabolism, yes, it is happening in our liver. Our liver having lots of that is substances which actually metabolize, like cytochrome P450. It can metabolize our drugs, also sometimes various kinds of food elements, but biotransformation is slightly different. Now, the biggest question, what is or what are the types of biotransformation? I hope it is very much clear to you all. Here I have written two types, enzymatic and non-enzymatic, because at the beginning of my lecture I have shared with you that we are actually using some bacteria, some fungi or enzyme. Some of the cases bacteria are also producing some enzyme. Either you can use those enzymes or either you can synthesize some enzyme in your lab, then you can use those enzymes for biotransformation, okay. Or otherwise in non-enzymatic, where we are actually not using enzyme, directly we are actually using some organisms. Okay. Now, what is enzyme, sir? What is enzyme? Enzymes are nothing but protein. Enzymes are some protein. In that protein, there will be some active site and there are chances that there might be some allosteric site, Okay. So, this is up to you. So, when you will design some project for biotransformation, you have to understand that how these things are actually arranged. When you select an enzyme, you have to check that substance. For example, I have a substance like this. Okay. I have a substance or metabolite or some substance like this. So, I have to check the active side of my enzyme and its active side, whether both are things are actually matched or not. Okay, let me tell you with a different color. Yeah, the active site of my enzyme and the component of our this substance, what is supposed to be metabolite, what is supposed to be biotransform, whether they are complementarily, they are actually matched with each other or not. If it is matched, then you may use that enzyme. Right now, we are actually using widely bioinformatics tool. Lots of bioinformatics tools are actually used for these purposes. Like for example, molecular docking. If you have 10 different kinds of enzymes and you have one substance and you want to check which enzyme will very nicely fit with that particular substance, you can do molecular docking. It will give you the result, it will tell you that which enzyme can be used. Right now, scientists are actually doing such kind of activity. Now, enzymatic, let me tell you about the enzymatic. So, this enzymatic are also divided into two parts. One is the microsomal and another is the non microsomal. In microsomal, that is a type of reaction or type of biotransformation. So, in this case we have to understand that enzymatic elimination is the biotransformation occurring due to various enzyme present in the body. Okay. So, the microbial biotransformation is caused by enzyme present within the lipophilic membrane of smooth endoplasmic reticulum. Okay. And in non microsomal, the biotransformation happening involve the enzyme which are actually present within the mitochondria. Okay. There are two different types. This microsomal is actually happening because of the presence with the lipophilic membrane of the smooth muscles of endoplasmic reticulum, and here it is happening due to the presence with the mitochondria. Okay. This is about this something called enzymatic, we are actually using enzyme. Non-enzymatic types of biotransformation for highly active or unstable compounds taking place at physiological pH. Some of these are include like chlorazepate converted into the this thing called desmethyl diazepam, or mustine HCl converted into the this thing called ethyl nimonium, or atracurium converted into the ludanosine or quaternary acid. Hexamethine also hexamine also converted into the formaldehyde. These are the few examples of non-enzymatic biotransformation. Here it is actually happening due to the physiological pH. Okay. You should remember these things. Now, come to very interesting point. What are the advantages of microbial biotransformation? You have to understand that the process of microbial biotransformations can operate at natural pH, ambient temperature and atmospheric pressure, while chemistry often requires extreme temperature, like temperature and other things of this condition, which are not exactly environmentally friendly, or in many of the cases we are actually getting industrially or undesired products. Okay. This is a great advantage of the microbial biotransformation. Another thing, extreme pH, temperature, pressure may provide harmful effect. Okay. If you do some activity in the industry, if you are providing extreme pressure, pH, temperature, pressure, they may provide harmful effect. towards the personal operating harsh procedure or may also affect surrounding community in the areas. Then also residual specificity and stereospecificity of the microbial biotransformation process, which allows the production of a chiral product or racemic mixture. Can you see? Your pure chemistry is actually coming into this picture. So, these things actually only possible through microbial biotransformation. If I tell you more, the ability of microorganism like fungi, bacteria, these things produce large amount of biomass and great variety of different enzyme in a short time. Okay, very short time it will be required. The small size has far the largest surface to volume ratio ever living on the earth. Concerns regarding transformation of animal based diseases such as bovine, this thing called spongiform, this thing called encephalopathy, that is BSE, or scrapie, kuru, or other kinds of various diseases we actually using to get the growth hormone purified. Okay. Several individuals or also individual reactions can be combined with a single microbial that is reaction. If I tell you more, sometimes it is cheap to use microorganism to prepare an organic compound then synthesize it chemically, and also production of the noble metabolite. So, can you imagine, for example, I hope many of you are doing B Pharm or pharmacy, you have learned about something called chain reaction. You have learned about the chain reaction. In your organic chemistry, your teacher might be teaching you about the chain reaction. For example, let me take one hypothetical scenario. There is some product, its structure is like this, and you want by doing bioinformatics based tools like docking and other structure activity relationship or QSAR activities, you have identified that there should be such kind of some enzyme should be there which will be able to bind with the substance, and after that it will be get degrade, it will be transformed into another desired product DP. So, now this has to be synthesized. There are two ways, either you can use the organic chemistry process or either whatever I have told you, that is a microbial biotransformation process you can use. Now, in organic chemistry, what you will do? You will be going with the chain reaction from A to B, B to C, C to D, D to E, and later on, for example, you got some molecule which have a similar structure and it can bind and it can degrade. But from A to B, you have used some that is a substance here, I am writing as in 1, here I am writing 2, 3, 4. You have used 4 different chemicals. After that, you got your ultimate desired product called E, and that thing work similar to this, and this is actually able to that is a bind with the certain protein, and it may degrade or it may create your desired product. But in all the 1, 2, 3, 4, 5 reaction, you have to give some time. Let me write. You have to give the time, you have to use the manpower, then you have to invest money, and sometimes it might be harmful because lots of chemicals are actually involved. Now, it will take a lots of time and you have to give a lots of efforts, but if you use microorganism, for example, you identified that there is some certain proteins actually present and you can use some enzyme, or you can use some bacteria, or you can use some fungi, and you have found these things can make your ultimate similar kinds of product. Okay, only one step is here, only one step is one protein or it may be a lipid or anything. Okay, this is just a one step process, you have to use some enzyme or fungi or other thing, and ultimately you are getting this desired product, this substance which will be doing a further activity. So, you can just compare, when you just compare your organic chemistry, let me take another color, you can just compare your this organic chemistry with microbial biotransformation. That is why in maximum cases we actually prefer, we actually suggest that you may go with the biotransformation by using the microbes, but still we have to work more on this field actually gaining popularity. In my next lecture I am going to tell you lots of application how we can use microbial biotransformation. Okay. Now, yes, everything having lots of advantages, but everything coming into the world with lots of disadvantages. What are these? First disadvantage is that the necessary expenditure for the development of biotransformation process, including the production isolations are actually quite usually high. The most causes of the reaction time rather long. The substrate or product concentration are actually low and the stability of the biocatalyst is actually limited. See, ultimately whatever the things actually are getting here, what are the product are actually getting, there are chances or there are sometimes there will be problems with this stability. There are I hope you might have studied the accelerated stability studies, and you also might have studied the its stability in environmental factor. So, you have to store it in a proper manner. For storage again you have to spend money. So, there are lots of issues might be there are chances. So, that is why everything having some advantages and everything having some disadvantages also. We have to take care of such disadvantages, and if you have to minimize these disadvantages, then this thing will be work very nicely. My dear students, as all of you are learning and all of you are very young and this field is rising, I will suggest that if you try to, if you have some time, you may spend your that is your free time by just doing or searching on Google about microbial biotransformation in 21st century. In US, China, Japan, Korea, Singapore or European countries, they are widely using microbial biotransformation to get their desired product. They are not relying on synthesis, that is organic chemistry by synthesis. So, yes, right now due to lack of technology there might be some disadvantages, but you can minimize those disadvantages if you give more and more research, if you spend more and more time to get new technology or new way of working. Okay. Now, where we are actually using this kinds of that is biotransformation, we can actually use in the transformation of various kinds of steroid and sterol. We also use biotransformation for the transformation of non-steroid compound, also transformation of various antibiotic, also transformation of various pesticides, pollutants and also petroleum biotransformation. Can you see? So, almost every area it is actually almost covered. So, we are actually using on those areas. So, ultimately I am on the way at the end of my lecture. I hope all of you have enjoyed the entire lecture, and today we have learned about the introduction of microbial biotransformation. You have seen what are the various types, that is two types, enzymatic and non-enzymatic. You have seen what are the advantages, you also have seen what are the disadvantages, and then at the end I have shown you few of the applications. In my next lecture I am going to tell you more about the application, how we can use those that is those substances, how we can make or how we can transform something to get our that is or we can transform our these substances to another product. So, all the things we are going to learn in our next lecture. So, thank you all of you for joining with me in today's lecture. I hope this lecture might have helpful for you for your examination and in your future studies and your research. So, keep learning, keep shining, and always be attached with our SWAYAM portal, and all the lectures will also available in all the respective platform. So, thank you all of you for joining. In our next lecture, we will learning something more. I am going to tell you the applied part of microbial biotransformation. So, God bless you all. See you in our next lecture. Thank you.